नहीं पियूंगा, मुझे लगा मिल्क नहीं है। तुम चाय पियोगे? पी लो, पी लो तो ले ले। ठीक है। So, a very good evening, everyone. A very good evening, everyone. So, we'll just wait for 2 minutes for the people to join and then we will start the session, okay? Okay, so we'll start the class now. So, a very good evening, everyone. Today, we'll be covering all the uh, components that were covered in week 10 lecture. So, week 10 lecture was about dosage compensation, cyst and non-coding RNAs in imprinting, particularly focusing the role of YYY. So, in the last class, we have learned about dosage compensation phenomena. So, let us quickly revise what is the meaning of this dosage compensation. So, in females, there is XX when we talk about humans, and males have XY chromosome. This means that females have two X chromosome. There is one extra chromosome. X extra is there. So, which means that the expression of X will be twice in females as compared to males. The expression of genes coming from X chromosome will be twice in females as compared to the males. Now, there will be an unequal expression of the genes related to X. So, to compensate this, nature has devised itself in such a way that one of the X chromosome does not share its expression. Expression comes only from one of the X chromosome. So one of the chromosome gets inactivated, and this phenomena is known as X chromosome inactivation. So what is the meaning of X chromosome inactivation? X chromosome inactivation means that one of the X chromosomes of the female gets inactivated and it gets converted to bar body. Okay. So one of the X chromosome is inactivated and it gets converted to bar body. So now the expression of X from male and female becomes equal because the expression is coming only from one of the X chromosome. And this inactivated X chromosome mostly is found in the, where it is present, if we talk about spatially, where this one inactive X chromosome would be present, this inactive X chromosome would be in the heterochromatin region. Okay, in the heterochromatin region, and spatially it is present in periphery. So this is present heterochromatin region, it is present in the periphery. So if this is a nucleus, this is the center, and this is the periphery of the nucleus. Okay. So genes which are active, which are actively transcribed, those genes are present in the center of the nucleus. And genes which are inactive, those are present in the periphery of the nucleus. So there is not only in the expression levels, I mean, not only in the condensation levels, but also the spatial arrangement of the gene also differs, that defines whether the gene is active or the gene is inactive. Active genes are present in the center of the nucleus. Inactive genes are present on the periphery of the nucleus. And this spatial organization is cell and developmental stage specific. This is cell and developmental stage specific. What is the meaning of this cell and developmental stage specific? This means that it's not like some certain gene X, suppose there's a gene X, it in all the cells it will be present in the nucleus, or in every developmental stage it will be present in the nucleus. No. In some cells it can be present in the nucleus, in other cells it can be present in the periphery. And likewise, for example, if you take the example of the gene which is responsible for the hair color, those hair color gene will not be expressed in the cells of liver because there it has no use. It will be expressed in your hair follicles, say hair follicle cells, but it will not be expressed in your liver cells. Why? Because liver cells, there is no use of the hair color gene. Okay, right? So all the genes are present in all the parts of the body, but the expression is cell and developmental stage specific. That means that all the genes are present in the DNA, but which genes will be transcribed, it depends on its cell and its developmental stage. So the genes which are actively transcribed, those genes are present in the nucleus. Genes which are repressed in that particular cell and developmental stage, or which are inactive in that cell and developmental stage, will be present on the periphery. So this is the cell and developmental stage specific expression of genes. So here when we are talking about the process of dosage compensation, there are two X chromosomes in females. One of the X chromosome which is inactivated, we call it as XI. XI means inactivated X chromosome. It is present as heterochromatin and it is present in the periphery of the nucleus. Periphery of the nucleus, why? because since it is inactive now, so is no need of it to be present in the nucleus. So it is present in the periphery of the nucleus. So I hope this is clear to everyone. Now, let us come to the first lecture for the week 10. First lecture was about dosage compensation, cyst and non-coding RNA in imprinting, the role of YY1. So what is YY1? YY1 protein, YY1 is known as yin yang 1. Okay, so basically we have studied that for the dosage compensation to happen properly, we require a non-coding RNA, which is a long non-coding RNA, and the name of that long non-coding RNA was ZIST. Okay, and this non-coding RNA was regulated by another um long non-coding RNA, which was T6, just the opposite of this. Now, this non-coding RNA does it work with the help of another protein, and the name of that protein is YY1. So this YY1, it kinds of helps cyst to perform its function. Now, how this YY1 helps cyst to perform its function? Because this protein helps in tethering the cyst onto the chromosome. That means the place where it is being produced. So we know that in the in the chromosome, wherever cyst is present, cyst gene is present, this has to localize in that particular part. Okay, this has to localize in that particular part. How cyst can be tethered or it can be attached or localized to its chromosome? This is done with the help of the protein, which is YY1. So YY1 actually helps the cyst non-coding RNA to be in its space, to be in its place. So if this is the chromosome and this is the place where cyst has to be there, so this YY1 helps the cyst to be there. Okay. So if YY1 is only blocked, then what will happen? Try to understand this. See, YY1, it helps in the localization of cyst. If YY1 is only blocked, then will this localization will happen? No, this localization will get inhibited. Why? Because the protein which is responsible for its localization is only blocked or only inhibited. And this, if this is blocked by some siRNA mechanism, then this localization is also affected. So that is what they are saying, that if you use YY1 siRNA, YY1 siRNA will block YY1 protein. To block its translation, then you will not have the ability of the cyst localized onto the corresponding chromosome. Yes, cyst will be able to attach to its targets, that is X inactivation center, with the help of the protein YY1 only. That means to perform its function, cyst needs YY1. And in the class, sir has also mentioned a paper, and in that paper it was mentioned that the mutation rate is seen to be very very high in the inactive X chromosome. So just I wanted to take 2 minutes to discuss this point, that we all nowadays we all see how much there is a spread of cancer or tumors, and the most of the cancers that we see that there is metastasis that is there. That means the cancer has spread from one place to another place. Okay, that is what metastasis means. So basically cancer happens because there is certain mutation that happens in the body, and that mutation causes the uncontrolled proliferation of cells. So normal cells, they show the property of contact inhibition. That means when they come in contact with a surface, they start they stops dividing. But cancerous cells, they do not show the property of contact inhibition, and they keep on dividing and dividing and dividing. So that is what cancerous cells have uncontrolled proliferation ability. Now, this is what cancer is about. But our body has made itself in such a way, our body tries its best to negate the possibility of any kind of mutation that can happen. Okay, so what happens that if there has to be a mutation also, it happens in the inactive X chromosome so that it is not affecting the body much, because inactive X chromosome is not contributing uh, for it's not I mean the genes in the X chromosome of inactive X chromosome are not contributing in the expression at all. So even if there is a mutation in the inactive X chromosome, it's not going to affect the body's expression of proteins or whatever it is. So this nature has defined itself in such a way, nature has made itself in such a way that most of the mutations, it happens in the inactive X chromosome, so that the active X chromosome is untouched, it remains as it is. Okay, so this is a paper that describes that. Now, yeah, this is what was there in the paper, that the mutation rate is seen to be very high in the inactive X chromosome. Okay, inactive X chromosome. And the rare mutation is possibly not different from autosomes, but the X inactive or inactive chromosome is just not repaired due to its late replication. Okay, that is not important point. So this is what is important point from the gist of the paper. Okay, so the inactive X chromosome, they accumulate somatic mutations during tumorigenesis. You see, this is a normal cell, this is a cancerous cell. So if you see, they did the sequencing, whole genome sequencing of a normal somatic cell also and a cancerous cells also. So you can see in cancer, the X chromosome has 4x more mutations than the autosome. Okay. So which means, see here, this is the cancerous, this thing, uh, cancerous X chromosome. So what this is meaning to say is that cancer means mutations. Okay. But even if we compare those mutations are more in the inactive X chromosome as compared to autosomes. So if you know that there are two types of cells in the body, I hope you know this, germ cells and somatic cells. So germ cells are the cells which are responsible, like X and Y, which has X and Y, which is responsible for deciding the sex of the body and all the characteristics related to that. Okay, so germ cells means X and Y, and somatic cells are all other cells. So in even in the case of cancer, if there are, let's say, 100 mutations, you will see that 60 mutations are in the X chromosome and 30 mutations are in the autosomes. And in that X chromosome also, these are in the inactive X chromosome. So that nature is trying its best to reduce the number of mutations. That is what I'm trying to say. It is trying its best to reduce the number of mutations so that the body is least affected. But even then, the body gets affected. So you understand ki uh, nowadays, even though we see that cancer has become very, unfortunately, very common, but body tries to make it as uncommon as it can by pouring most of the mutations to the inactive X chromosomes. Okay, fine. Now, genetic imprinting and shRNA. So, what is the meaning of genetic imprinting? Genetic imprinting is a process which results in expression on only one allele of a gene, while the allele originating from the other parent is silenced. What is the meaning of this? So, for any gene, if you know that one gene has generally two alleles. One allele come from mother and one allele come from father. We know that it is a random process For example, if we talk about, let's say, skin color, okay, I will take the example of skin color here. Fine. Skin color. Now, skin color, for the skin color, allele, suppose mother's is a fair, mother is fair, and father is wheatish, wheatish complexion. Now, the progeny will can either have a fair color or it can have a wheatish complexion. It is random. Okay, I mean, whichever allele come, that color of the child will have. Either if the allele is coming from mother, he or she will have fair color. If the allele is coming from father, it will have a wheatish color. So here it is a random process. We don't know, but there are certain characteristics. There are certain genes which are pre-decided or predestined that they will come from mother only or they will come from father only. That means that gene is imprinted to be coming from mother or it is coming from father. That kind of phenomena is known as genetic imprinting. Okay, so genetic imprinting is a process which results in expression on only one allele of gene, while the elder allele originating from the other parent is silenced. Process is somewhat similar to doses compensation. The difference of expression from both alleles are due to different states of chromatin and also due to different methylation of DNA. So basically, whenever anybody is talking to you about expression, how much expression is happening from a particular allele or a particular gene, so always remember that it depends. Right. Okay, so it depends upon the heterochromatin or euchromatin state or in the methylation region. Activity of small heterochromatic RNA, which is known as shRNA, appear to be essential for establishing and maintaining the imprinting status. And activity of various shRNA not only limited to genetic imprinting. So there are two things to understand: what is genetic imprinting, the concept of genetic imprinting, and how genetic imprinting takes place. So shRNA helps in genetic imprinting. This is important. Okay, what is important is that shRNA helps in genetic imprinting. This is an important point. Okay, so now we come to the lecture. This was all about lecture 1. Now we'll come to lecture 2. So lecture 2 was about shRNA and gene imprinting. So we know that shRNA is responsible for genetic imprinting. Now, to perform its function, there are two different models that shRNA can perform. One is the DNA recognition model, and the second one is the RNA recognition model. Okay. Basically, these two models helps in the condensation of chromatin. Just now here we studied that genetic imprinting means one of the allele has to be silenced. Okay, one allele has to be silenced and it is already pre-decided whether this allele will be of father or mother. Suppose father's allele has to be silenced. Father. Okay, so now silencing means what? The chromatin has to be converted into a heterochromatin state. Silenced chromatin is always a heterochromatin. And what is the characteristic of a heterochromatin? That the chromatin is condensed. This is the property of heterochromatin, that chromatin is condensed. I have already discussed this concept in my previous classes also, that when chromatin is very tightly packed, okay, when the chromatin is very tightly packed, that it is highly condensed, then there is no space for the transcription factor to come, sit, bind, and perform its function of transcription. So, in order for the transcription factors to come, sit, bind, and perform its function, the chromatin has to loosen up a bit so that there is a place for the transcription factor to perform its function. So, therefore, if the chromatin is condensed, then it will automatically shift into heterochromatin state. And because it is so much condensed, that is why it is stained darkly, because one line versus this much line. So what, which is more darker? This is more darker now. So because it is, if these are the lines like this, and if the lines are like this, which will be darkly stained? This one, because it is more condensed. That is why heterochromatin is darkly stained. So now this shRNA, which helps in the genetic imprinting, it does its function by condensing the chromatin. Now for condensing the chromatin, it has two models. One is the DNA recognition model, okay, and the other one is the RNA recognition model. So let us try to understand what is the meaning of this DNA recognition model and RNA recognition model. In both the models, similar pattern is observed, except that in DNA recognition model, there is no need of gene expression, obviously, because it's a DNA, no? DNA expression of gene happens at DNA level or RNA level? Expression of genes always happen in RNA level. So when we talk about the DNA recognition model, which means that there is no need for the DNA to be expressed. Fine. And so it can be imprinted because of an shRNA coming from elsewhere. So this is an shRNA. It comes and it binds to the DNA, and which leads to the coming of some silencing factors, some modifying enzymes, and ultimately what happens? This DNA is silenced. That means this DNA will not go for transcription process. Could you understand what happens here? So what happens here is that shRNA from else, sorry, yeah, shRNA from elsewhere comes. It comes and binds to the DNA, which leads to the coming of other factors such as silencing factor, histone modifying enzymes, etc. And then this DNA is marked, and then this DNA will not be transcribed. So this is how this shRNA recognizes the DNA. But in the RNA recognition model, what happens? Little expression happens. But after a while, the shRNA will bind, and then there will not be any further transcription. Okay. So this gene has to be expressed for a while, and then this will be triggered by the shRNA binding, and it will lead to the silencing through the silencing complex. Okay. So it is like this. So I hope this is clear that how shRNA does its function. So shRNA has basically two models: DNA model, DNA recognition model, and RNA recognition model. The ultimate work of shRNA is to condense the chromatin, because if the chromatin is condensed, it will be converted to heterochromatin and it will not be active. Now to condense the chromatin, it has to come and bind to the chromatin. It has to find the chromatin and it has to find its target, and it has to bind there. So for that, it has two models. One is the DNA recognition model, other one is the RNA recognition model. In DNA recognition model, what happens? That shRNA from elsewhere comes and it binds to the DNA, and then other factors comes, and then there is the condensation of the chromatin, and then there is no this thing expression. In RNA recognition model, this gene is expressed just like this has to be expressed. That means shRNA is expressed here. It is not coming from outside. This shRNA is expressed here itself. And when it is expressed, then it binds to the target complex, and then it causes the inactivation of the gene. So here shRNA is coming from elsewhere. Here shRNA is expressed here only, and then it is performing its function. That is the difference between DNA recognition model and RNA recognition model. I hope this is clear to everybody. So now this was transcriptional regulation. Here also this DNA, it binds to DNA, and then there cannot be any transcription. Here also what happens? that there cannot be any transcription. What if transcription has already happened? Then can it not be prevented? Yes, it can be prevented. How it can be prevented? If even if transcription has happened, we can try to control it at the translation level, because whatever transcripts are there, ultimately those transcripts have to be converted to proteins, right? Those transcripts have to be converted to proteins, then only the function will take place. So we can control it at the transcription level, and if it is not getting possible to control at transcription level, we can also try to control at translation level. So what happens for translation levels? We have non-coding RNA, and we have a phenomena of RNA interference. Why we are studying all this? These are the different ways by which dosage compensation can be performed. Okay, we should see, while studying the flow of it, we should not forget why we are studying it. So we should know the reason why we are studying it. So we are studying it because these are the different mechanisms, uh, are the different ways in which dosage compensation can be performed. Fine. Now comes protein function modulation. Okay, see, the body has made itself in such a way that it tries till the last breath ki how we can control it. We'll try to control at DNA level. Okay, if it is not possible at DNA level, we'll try ki we can control it in the RNA level. That it will not transcribe. Even if there also we could not could not do, we will try at translation level. That okay, RNA has been made, RNA has expressed, chalo, at least translation will not take place, proteins will also not form. But now what if the proteins are also formed? Then it tries ki okay, chalo, protein has been formed, we will try to modulate its function so that the protein cannot perform its ultimate function, because the ultimate goal of the protein is to perform its function. So some non-coding by RNA can directly bind to proteins, altering their structures, enzymatic activity, or ligand binding. Targets of such non-coding or RNA often are proteins which are involved in transcription, for example, nuclear receptors or general transcription factors. So, see, some proteins, suppose if this is a transcription happens with the help of certain transcription factor, and if this transcription factor is only terminated, then this transcription will not take place. So this transcription factor can be inhibited by its binding to certain non-coding RNA. So non-coding RNA can bind to this protein, and then it can alter the function of this protein, and ultimately it cannot perform its function. So, see, this is a molecular biology course, right? So, hmm. So whenever you are studying a mol bio course, first thing that we make students study in a mol bio course is the central dogma. And then people say that this is such a boring thing. We all know DNA to RNA, RNA to protein, protein to metabolome. What is new in this? New, nothing is new, but the connections that you have to make while you are understanding the topics, that is important. That's why always central dogma, DNA to RNA, RNA to protein, and protein to metabolites. So this RNA phase and protein phase decides the expression of a particular gene. First, there is a gene is expressed RNA, the RNA is translated protein is used, and metaboloids are formed. This central dogma is very important, because this central dogma will only guide you whether if there has been a problem at the mRNA level, can it be solved at protein level, or can it be solved at some other levels? Okay, fine. So now RNA interference, we have already studied how RNA interference takes place. You have the preliminary RNA, which is acted upon by DROSHA and it is converted into pre-miRNA. Then this pre-miRNA, I'm talking about miRNA, the same is for siRNA also. It goes from the nucleus to the cytoplasm. In the cytoplasm, it is acted on by dicer, and then one is a passenger strand, the other is the template strand. Template strand goes and binds to the risk complex, and then it's perform its function. This is something that we have already discussed. This is the mechanism, biogenesis and mechanism of how non-miRNA or siRNA are formed. Let us try to see what are the functions of this RNA interference. So the first function is it functions as defense against double-stranded RNA containing viruses. So if there is any double-stranded RNA which has virus, then immediately it has to be stopped so that it does not get expressed. So this is its first function, that it acts as a defense against double-stranded RNA containing viruses. Then it stabilizes the genome by sequestering repetitive sequences such as mobile genetic elements. So if the genome has a lot of repetitive sequences, then that is also a problem. Okay, so it stabilizes the genome by performing the RNA interference activity on certain mobile genetic elements. Can you name any one mobile genetic element? Any mobile genetic element that you remember? So the mobile genetic element is transposomes that we have already studied. Okay. Then control cellular development. Dicer knockout mice do not survive past gastrulation. Okay. So it also control the development. If a mouse does not have dicer, it cannot perform its function. Past gastrulation, it cannot perform its function. Okay. Then, so this is just a quick reminder. I'll just put it in the slideshow. See, siRNA and miRNA, they have similar functions. So pre-miRNA, it is acted upon by dicer. Then one of the strand, passenger strand, and there is a template strand. So template strand goes, it binds to the risk complex, and then it performs its function. Then miRNA, again, it is acted upon by dicer. shRNA is also acted upon by dicer. Then active risk and mRNA cleavage. Now, miRNA also perform its translational repression and siRNA also perform its translational repression. So what is the difference between RNA interference and miRNA function? RNA interference, we're talking about siRNA. I mean, we're talking about, yes. And miRNA, we have already studied. So what happens here is that both are actually performing repression only. See, what I'm trying to say is that siRNA, it performs RNA interference and miRNA also performs interference. I mean, it is also involved in repression. But there is a small difference between siRNA mediated repression and miRNA mediated repression. What is that difference? That if this is the target and it is A, G, C, C, A, T, T. So miRNA for the non-coding RNAs to come and repress a particular gene, it has to first go and bind to it. Okay. Now miRNA, will not base pair with it perfectly. What does that mean? It can base pair like T, C, 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 T, A, A. So is it a perfect base pairing? No. Why? Because here G should be there. Here G should be there. So base pairing is there, but it is not 100% perfect. This is the case of miRNA. Whereas when we talk about siRNA, it will have a perfect base pairing. So it will be T, G है तो C, G, G, T, A, A. So this is siRNA and it's a perfect base pairing. So miRNA will not do perfect base pairing. Okay, I'm writing here. Very important to understand. Please pay attention to this concept. This is a very important concept. miRNA imperfect base pairing. Imperfect base pairing. I'm really sorry for my bad handwriting. I cannot help it. Whereas siRNA, it has a perfect base pairing. Now, what is the consequence of this perfect or imperfect base pairing? Because miRNA do not has a perfect base pairing, so it cannot cleave its target, but it will block the translation. Okay, it cannot cleave the target. Target cleavage, no. So miRNA cannot cleave the target, but it will block the translation. Whereas here it will cleave the target. Here there will be translation repression. I hope you are able to understand. If you have any problem, do let me know. So miRNA will thus cause translation repression because it has repression because it has imperfect base pairing. Imperfect base pairing. Whereas siRNA, it will cause target cleavage. Because it has perfect base pairing. Okay. So this is the difference between the mechanism which is used by miRNA and the mechanism which is used by siRNA. So I hope this is clear. Now, is RNA interference exclusively limited to cytoplasm and post-transcriptional control? Although this is a very common view, it does not always have to be the case. siRNA can be transported back to the nucleus and act as shRNA to block the transcription also. Okay, so always remember that mostly what happens, this uh, all this happens in the cytoplasm. Whether it is miRNA or it is siRNA, it comes out of the nucleus to the cytoplasm, where it is acted upon by dicer. Then it is converted into a template strand and passenger strand. Then it gets loaded to the RISC complex, and then it performs its function. So that means it is happening in the cytoplasm. But it is always the case. Most of the time it is the case, but not always, because sometimes siRNA can be transported back and it will act as shRNA. Okay. So now there are two models for genomic imprinting. First is the insulator model and second one is the long coding RNA model. Knowing things that genomic printing we have already discussed right now. Main thing which you should remember in these two models are that insulator model is employed by H19 IGF2 imprinted locus, and LNC model imprinting at the IGF2R locus, and which is actually also known as ICR, differentially methylated. And insulin model is older model, and LNC RNA model is a newer model, which is more commonly used model. That much only you have to remember. Then you can see what is the effect of genomic imprinting. The effect of genomic imprinting can be understood by taking the example of Hox clusters. So one set of transcription factors which is critical in embryonic development is encoded within the genes by Hox cluster. So what happens? What are Hox genes? So these Hox genes are a group of genes that control the body plane of the embryo along the anterior posterior axis. Following formation of the embryonic segments, expressed Hox protein determines the type of segment structures. So what happens? That this is the axis of a plane, axis plane of any organism. Hox gene will decide that this particular will be your head, this would be your tail, okay, here would be your wings. So whatever it is, whatever the segmentation is done, it is done by Hox genes. So Hox genes are very much important because it kind of decides the whole way in which the organism will look. Where will be the head? Where will be the wings? Where will be the tails? Where will be the antennas? Everything is decided. So if there is any problem in Hox gene, what will happen? There will be malformations. In the place of head, there can be tail. In the place of tail, there can be head. So a malformed body would be caused if there is any mutation in the Hox gene. Now, Hox gene clusters are encoded by long coding RNA genes, hot air, hot tip. This is important. So I'm just telling you the important points related to your examination point of view. Okay, this is this is important. Okay. Hmm. Then the LNC RNA hog and TOG are encoded by two genes. This is important. And the function of Hox genes that it controls the body plane along the anterior posterior, that is head to tail axis. Legs, antenna, wings, all this is important. So I have just marked the important points from here. Okay. I hope this is clear. So now these are just the description of the various uh, long coding RNAs which are present. Hot air, then hawk dog. You can just read it. This is just like the reading part. I'm not going into very much detail. Then homeotic transformations. What is the meaning of homeotic transformation? Can anybody tell me? What is the meaning of homeotic transformation? Homeotic transformation means the change in the structure with another structure. A given place, a structure should be there that is missing. Suppose, see, this is a wrist and some kind of malformation has occurred. So the structure, the wrist is not properly formed. A malformed body, head not properly developed, tail not properly developed, fingers not properly developed. So a malformed body, that is your homeotic transformation because of the Hox genes. Okay, then you see that now we'll be seeing the different roles of long non-coding RNA. So there are many different roles of long non-coding RNA. Lecture 5 is about different non-coding RNA and their roles. So non-coding RNAs are involved in muscle differentiation. For example, it controls the transcriptional factors such as PAX5 and all that. Then non-coding RNAs are also involved in retinal development. So you need not to go into each and every details of it. Okay, I'm just trying to tell you how much is important. You should just know that what are the different roles of non-coding RNA. So first is, it is involved in muscle differentiation, it is involved in retinal development, it is involved in some kinds of the non-coding RNA also has a role in disease. Okay, uh, for example, Alzheimer's disease. Long non-coding RNA is involved in Alzheimer's disease. Then hot air is also involved in diseases. Mir 200 is involved in cancer. So, which means that these non-coding RNA is involved in complex diseases as well. Then long non-coding RNA induced cancer. So there are so many different non-coding RNAs which causes and the progression of cancer. Just to read about, there is nothing much to explain in here. Then Terra. Now this is little bit I will explain. Terra means basically telomerase is the enzyme which is responsible for the forming of telomeres at the end. Okay. So what happens? Telomerase enzyme. Okay, I will tell you like this. If this is your cell has telomeres at the end. I mean, genome has your telomeres at the end. With each round of replication, this telomeres starts to getting shortened up. So if this is a cell, telomeres, after one division, it will become this one, this much. Then second division, this much. Third division, this much. Fourth division, this much. Fifth division, completely. So after fifth division, this cell cannot go. And after fifth division, this cell will die. Because eventually telomeres get shortened down. So what happens? That cell is dying because the telomeres are getting shortened. But we know that cancerous cells have a property of uncontrolled proliferation, and they do not show the property of cellular senescence. That means they do not die. So if they do not die, this means that their telomeres are constantly getting refreshed. Constantly new telomeres are there and they are getting added up at the ends. है ना? Because telomeres are responsible for keeping a cell alive, and since after each round of replication, telomeres get shortened up, which means that the cell will eventually die. But in the case of cancer, the cell is not dying because telomeres are constantly getting renewed. So if the telomeres are constantly getting renewed, so the cancer cells will not die. So in this case, Terra, TERRA, what it does is that it binds with the telomerase enzyme and it inhibits its activity. So telomerase is responsible for making the telomeres. Telomerase is the enzyme which is responsible for producing telomeres at the end. If we inhibit this enzyme telomerase, what will happen? Telomere renewal will get stopped. So that is what they are saying, that if this Terra is down regulated, it increases the cancer cell longevity. So LNC RNA transcribed from telomere ends, which can bind to telomerase enzyme, inhibiting the activity, and it is down regulated. So Terra is a LNC RNA, and this non-coding RNA is down regulated in cancer cells, because of which the telomerase activity is high, enabling the cancer cells to evade apoptosis or cellular senescence and subsequent programmed cell death. So the gist of this paragraph is that Terra is a long non-coding enzyme, a long non-coding RNA. And the function of this non-coding RNA Terra is that it goes and it binds to telomerase enzyme and it inhibits the activity of telomerase enzyme. And when it inhibits the activity of telomerase enzyme, telomeres formation at the end is decreased, and therefore the cell will die eventually. But in the case of cancer, this Terra is inhibited, And because this tera is inhibited, telomerase keeps on making telomeres, and therefore the cell doesn't die. The cells keeps on proliferating. Is it clear to everybody? The function of tera, what I'm trying to say? That tera is a long non-coding RNA which inhibits the function of telomerase enzyme. This is what it means. Now, this is about how cyst is involved in cancer, not to go in very much detail. And what are telomerase? I already told you that telomerase are the enzymes which are responsible for production of the telomeres at the end. Okay, now we'll come to the questions. Okay, so we'll do the questions as we already always do. I will put the question on the screen. I will give you 2 minutes. You think about what can be the correct answer. And after 2 minutes, I switch the screen and we discuss the question. So the first question is, non-coding RNA is not involved in development, transcription elongation, differentiation, and imprinting. So in which of the following phenomena you think non-coding RNAs are not involved? Right now it is 7:53. I will switch the screen after 2 minutes. Okay. So I will switch the screen 7:55 approximately. Think and try to answer. Okay. 1 more minute with you. Then I'll switch the screen. Okay. So what would be the answer? The question is, non-coding RNAs are involved in development, transcription, elongation, differentiation, and imprinting. So the correct answer is transcription, elongation. It's not involved in. Why? Because it is involved in development. Yes, it is involved in differentiation. It is involved in imprinting also. It is not involved in transcription, elongation because non-coding RNAs generally try to control a process, and transcription, elongation uh, 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 mostly protein current. So the correct answer is transcription elongation because it controls the development, it controls differentiation, imprinting we have already studied, but we haven't studied any example of transcription elongation because it's a positive process and non-coding RNA generally controls or inhibit the activity. So we are not in elongation, we do not want any inhibition, we want the transcription to take place. Then the next question is, homeosis refers to this is we have discussed formation with wrong structures, formation with malformed structures, formation with broken structures or formation with big structures. Right now it is 7:56. After 2 minutes, I'll switch the screen and I will ask you. 1 more minute left. Think. Okay. Now I am switching the screen. Hmm. So the question is homeostasis refers to, the correct answer is formation with malformed structures. We have already studied that Hox genes are the genes which control the body patterning during development. Okay, it controls the anterior posterior axis, where head will be formed, where tail will be formed, where are the wings. So body patterning. Body patterning is controlled by Hox genes. And if there is any mutation in the Hox genes, then what happens? There is formation of malformed structures takes place, and therefore homeosis happens. So homeosis is a phenomena which describes the formation of malformed structures. Then next question is, which of the following RNAs do not contribute in human disease? miRNA, siRNA, rRNA, or LNC RNA? Means which of the miRNAs are not involved in any diseases, are not linked with any diseases. Right now, it is 8 o'clock. I will switch the screen after 2 minutes. Okay? One more minute left. Okay, now I'm switching the screen. Hmm. So the question is, which of the following RNAs do not contribute in human diseases? So the correct answer is ribosomal RNA, because ribosomal RNA is just is it is involved in protein synthesis, and it is not directly known to play a role in any disease mechanism. Whereas when we talk about miRNA, we talk about siRNA, we're talking about LNC RNA, which is a long non-coding RNA. So these are basically involved in the regulation of various phenomena which ultimately translates to diseases like cancer. We were studying many kinds of neurodegenerative diseases, also genetic diseases, like see here, this non-coding RNA is just, it is involved in cancer. Then so many diseases are involved. Alzheimer disease is there. Emirit 200 is involved in cancer. Then there are certain retinal development, that is also one of the areas where it is involved. It is also involved in muscle differentiation. So because they are involved in different phenomena, cell metabolic phenomena and cellular phenomena, and each phenomena is linked to some kind of a disease. So here it is written siRNA. The correct answer is rRNA only. Okay, the siRNA is wrong. Correct answer is rRNA. Now, hot air is a non-coding RNA, structural RNA, coding RNA, or a sponge RNA. Hot air, which kind of RNA is it? Right now it is 8:04. I will switch the screen after 2 minutes. Think we have discussed this point already. One more minute left. Okay, time's up. So have you tried answering it? No? Okay. So hot air is, what is the correct answer? It is a non-coding RNA. We have already studied here. See? Huh. Hox cluster. Hox genes are a group of related genes that control the body plane of the embryo. That's why I have marked it as important, because many questions are getting framed from this particular topic, and it is anyways it is important even if you're trying to give any competitive exam. So in your competitive exams also, this is an important topic. So that is why I have marked this as important. So Hox genes are a group of related genes that control the body plane of the embryo along the anterior posterior axis. And this Hox is actually a cluster of genes. Hox genes are a cluster of genes, and these includes long non-coding RNA LNC RNAs such as hot air, hot tip, hot air M1, and these long non-coding RNAs have different functions also. For example, hot air, which stands for Hox antisense intergenic RNA, it is transcribed within the Hox6 cluster, and it regulates Hox gene in trans by repressing the genes, whatever the function is there. No need of going into that. Just remember that hot air has a trans regulation. Hot air is involved. Okay, fine. So the overall gist is that hot air is a long non-coding RNA. Okay. Then next is dicer acts in cytoplasm, nucleolus, nuclear pore complex. Let us take it as nucleus. I don't know why they have given the same option. Okay. Right now it is 8:08. I will switch the screen after 2 minutes. Meanwhile, you try to answer. One more minute left. Okay, the time is up. Okay, I'm switching the screen now. Okay. Have you tried to answer this? No? Fine. So, dicer acts in cytoplasm. We have already discussed it that in the cytoplasm, drosha and dicer are the two things that students get confused in. So, drosha it acts in the nucleus, and dicer, it acts in the cytoplasm. And then the passenger RNA and template RNA, two things are formed, and they go and then they act on the risk complex, and then they perform its function. They are loaded into the risk complex and then they perform the function. So the correct answer is cytoplasm. Next question is, RNA interference was first discovered in A. zebrafish, B. C. elegans, C. Saccharomyces cerevisiae, or D. Saccharomyces spongium. What is the correct answer? Right now it is 8:12. I will switch the screen after 2 minutes. 1 more minute left. You did some still short. Okay, now I am switching the screen. So the correct answer is C. elegans. So RNA interference was first discovered in C. elegans. There's nothing much to explain this question. Then next question is, any gene mutation results in homeosis, true or false? Any kind of gene mutation will result in homeosis. Think. 2 minutes. Right now it is 8:14 only. I will switch at 8:16. 1 more minute left. Okay. Switching the screen now. So the answer would be false. Why? Because homeosis is a phenomena which results in the formation of malformed structures. Okay, malformation. There is malformation of structures. So malformation of structures is caused because there is a mutation in the Hox gene. Hox gene. There is mutation in the Hox clusters. Okay, so not all gene mutations will result in homeosis. Homeotic transformations specifically occur due to mutation in Hox genes or genes which are involved in body patterning or segment identity during development. Okay. And mutation in other types of genes may lead to different effects such as metabolic disorders, cancers, or other genetic diseases, but it will not necessarily it not be homeosis. So it is not like mutation in other genes will not result in any kind of problems. No, they will also result in some or more kind of problems, but those those would not be homeosis. It can be any other thing. It can be some genetic diseases, some metabolic disorder, some kind of cancerous conditions. But if we are talking specifically about homeosis, then homeosis occurs because of the mutation in Hox genes. Okay. Now, next question is, sense and antisense transcript often act antagonist to each other. True or false? Okay, this is actually a good question. So right now it is 8:17. We will switch the screen after 2 minutes. And we'll see. One more minute left. Okay, now I am switching the screen. Hmm. So, sense and antisense transcript often act antagonist of each other. It is true or false? It is true. So, sense and antisense they regulate their gene expression. So, how they can regulate their gene expression? They can regulate gene expression by acting antagonist of each other. Okay. So, antisense RNA can block translation, it can induce degradation, or it can alter the splicing of sense transcript, which is leading to gene silencing or modulation of gene expression. And this mechanism is commonly, you can see it is all observed in gene regulations, imprinting, disease process. So sense and antisense are complementary to each other and they act antagonist to each other. So it is true. Okay. Next question is, genome imprinting is not seen in all living organisms. Is it true or false? First of all, you should know what is genome imprinting. We have already discussed what is genome imprinting. That certain alleles are silenced and it is already imprinted that in for a particular trait, the allele of the father would be silenced or would be imprinted, or the allele of mother would be silenced and father would be imprinted. So that is already decided. So that is your genome imprinted. So the silencing of the other one of the uh, 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 paternal or maternal gene, that silencing is somewhat related to dosage compensation, where there is condensation of the chromatin which takes place. This is something which we have already discussed. So accordingly, what should be the answer? Genome imprinting is seen in all living organisms. All organisms will include everything. That means it will include bacteria, fungi, everything. Think. Right now it is 8:21. I will switch the screen after 2 minutes. Okay. Okay, 1 more minute left. One more minute is there. Okay, now I'm switching the screen. So, is genome imprinting seen in all living organisms? False. Why? Because imprinting is primarily observed in mammals and some of the flowering plants, and it is absent in most of the organisms. Okay, so like I gave you the hint also. For example, we have bacteria, we have fungi, and we have so many invertebrates. So, do you think that they show genome imprinting? No. So, imprinting involves uh, it is involved in mammals and some of the flowering plants. And genome imprinting, as I told you, that alleles of one of the parent would be expressed. It is already known, it is already decided, it is imprinted that maternal alleles will be carried forward or paternal alleles will be carried forward. And this imprinting involves epigenetic modifications such as DNA methylation that leads to parent specific gene expression. Okay. So imprinting involves epigenetic modification. It involves epigenetic modifications. Okay. Next question is shRNA could participate in chromatin condensation. Already discussed. You should be able to answer this. True or false? Right now it is 8:25. I will switch the screen after 2 minutes. Think. Think and try to answer this question. One more minute left. Okay, I'm switching the screen now. Hmm. Hmm. So the correct answer is true that shRNA it participates in chromatin condensation. How shRNA participates in chromatin condensation? Basically, shRNA leads to the formation of heterochromatin structures. And it leads to the formation of heterochromatin structures through histone modifications and DNA methylation, which contributes to ultimately gene silencing. So basically, it shRNA It leads to the formation of heterochromatin structures. Okay. And these heterochromatin structures are formed because of certain histone modifications and DNA methylation. That means epigenetic changes that takes place. Epigenetic changes that takes place, such as histone modifications and DNA methylations. Okay, so this is about the question. Now quickly we'll try to revise whatever we have studied today. So week 10 was about, this was in continuation with dosage, compensation, cyst and non-coding RNA in imprinting and the role of YY1. So what is YY1? YY1 is basically the protein which is required or responsible for the localization of the cyst non-coding RNA, which ultimately helps in the dosage compensation. Then we have studied about that mutations that happens if there is an active X chromosome and then there is an inactive chromosome. So most of the mutations, they are uh, uh, they take place in the inactive X chromosome. This is what we talked about. Then we talked about genetic imprinting and the role of shRNA. So basically genetic imprinting is undertaken by shRNA. Okay. So what is genetic imprinting? First of all, genetic imprinting, it is a process which results in expression of only one allele of a gene, while the other allele originating from other parent is silenced. And this process is similar to dosage compensation, and this takes place with the help of small heterochromatic RNA, which is known as shRNA. Okay. So how shRNA does it function? There are two models, DNA recognition model and RNA recognition model. So basically shRNA causes the condensation of chromatin. And because of the condensation of chromatin, there is formation of heterochromatin structures. And then that particular gene is, that particular is silenced. So DNA recognition model, externally shRNA comes and binds to the DNA. And then there is a addition of several other factors, histone modifying enzymes and methylations, etc. take place, and it is silenced. Whereas in the case of RNA recognition model, what happens? That shRNA is transcribed by the RNA and then it silences the gene. This we have studied. Then we have studied that there can be translational regulation also, or there can be protein function modulation also. And then we have studied about what are the different functions of RNA interference. So it helps in like defense against double-stranded RNA viruses. It stabilizes the genome. It controls the cellular development. So these are the different functions of RNA interference. Then we have studied very important how the work of miRNA is different from siRNA. So miRNA does not have a perfect base pairing. miRNA has imperfect base pairing, whereas siRNA has a perfect base pairing. So because miRNA do not have a perfect base pairing, so it cannot cleave the target. It cannot cleave the target RNA. It only causes the repression of translation. So there is translation repression. Whereas siRNA, because it has a very perfect base pairing, it causes the cleavage of the target. So this is how the function of miRNA is different from siRNA. Both are involved in repression or silencing of the genes. But the action or the way by which they perform its activity is different. Okay. Then, is RNA interference exclusively limited to cytoplasm and post-transcriptional control? No. Although mostly it happens in the cytoplasm, but there is shRNA which can go again from the cytoplasm to the nucleus and it can block the transcription process. This is what we have studied. Then genomic imprinting. What are the two different models of genomic imprinting? There are two models of genomic imprinting. Hmm. There are two models of genomic imprinting. One is the insulator model, other one is the LNC-RNA model. So this is what we have studied. Then we have studied about the Hox genes, how Hox genes are involved in a mutation in the Hox genes are involved in body patterning, and any mutation in the Hox gene is involved in homeotic transformation. And these Hox clusters are nothing but the long coding RNA genes. Okay. Then we have studied the different roles of non-coding RNA. So they are involved in muscle differentiation, they are involved in retinal development, they are involved in various kinds of diseases like cancer, Alzheimer's disease, many different kinds of diseases they are involved in. Then they are also involved in cancer, particularly cyst is involved in cancer. And then we have studied about a long non-coding RNA, and the name of that long non-coding RNA is TERA. And what does TERA does? TERA, it binds to the telomeres and it inhibits the activity of the telomerase enzyme. And because it inhibits the activity of telomerase enzyme, what happens is that telomerase is inactive. So the telomeres at the ends are not renewed, and therefore the cell ultimately dies after certain rounds of replication. But in the case of cancerous cells, what happens? This TERA is down regulated. Okay, this TERA, it is down regulated, and because it is down regulated, the activity of telomerase enzyme is very high, which enables the cancerous cell to evade the apoptosis process or cellular senescence, and subsequently program cell death also. Then we have studied that cyst is very much involved in cancer. I mean, not much to explain over here, but yes, cyst is involved in cancer. And then we have studied what are telomerase enzymes. Okay, what are telomers? So basically, telomers are what? These are the repetitive DNA sequences. At the end, if this is a chromosome, at the end you have some sequences which are repetitive sequences. And these are present at the ends, and these are known as telomeres. They contain thousands of repeats of 6 nucleotide sequence TTAGGG. So this is the sequence of telomere. It is important. TTAGGG. I think I forgot this slide. TTAGGG. This is the sequence of telomere, and this sequence is repeated thousands and times. And what happens that these telomeres actually protect the chromosome from replication related loss of vital genetic information. That after each round of replication, it can, if there are no telomeres, what will happen? The main DNA part, it will start getting um, mm, mm, degrading. If this is this after second round, one round of cell division, it will become this, then it will become this, then it will become this, then this. So what is happening? This is DNA with no telomeres. So there is a loss of genetic information, isn't it? So to protect this loss of genetic information, there are telomeres. So after each round of division, there is a loss, but not of the main DNA, of the telomeres. So telomeres are getting lost, but not the main DNA is getting lost. So DNA, by losing itself, is protecting the main DNA from any kind of loss. So in this way, telomeres are actually very important for maintaining the genetic stability. Because if telomeres are there, then only the correct information and the full information is passed from the parent to the progeny. If telomeres are not there, so telomeres are important. Do not think that telomeres are not important. The only problem is that anything beyond a certain level is bad for us. Okay, so telomeres are important, but if there is uncontrolled division and there is a lot of telomeres so that the cell will not die anytime, then that is a problematic situation. So in cancerous condition, what happens? That there are genes and some are pro-oncogenes. What is the meaning of pro-oncogenes? Means those genes are involved in proliferation. Any gene which is involved in proliferation is not a bad gene, because proliferation is a basic requirement of human body. So proliferation genes are required. But if there is uncontrolled is the word which has to be used every time when we are talking about cancer. If there is uncontrolled way of anything, uncontrolled replication, uncontrolled proliferation, uncontrolled secretion of certain thing, then that is a problematic situation. So everything has to be there, but in a certain amount, up till a certain time. If it goes up till even after that, then that is a problematic situation. So it has to be a balanced diet. For example, in our body also we need sugar, but too much of sugar will again lead to many different disease conditions. We need protein also, but too much protein can also lead to some kind of uric acid concentrations in the body. So in short, everything is required in the body, but in balanced amount. So telomeres are also good. They are actually important because it protects our chromosome and it protects the important genetic information. But if there is uncontrolled proliferation of telomeres because of the enzyme which is telomerase, okay? So there are two words, telomere, telomere, which is the repetitive sequence, and telomerase. Okay, so telomerase is an enzyme. So ase, if you know, ase is the suffix, and ase is the suffix which is used for describing any enzyme. So if there is ase, this means it is an enzyme. Okay, so telomerase, if there is uncontrolled proliferation of telomeres because of the telomerase enzyme, then that is also a problematic situation. Therefore, to regulate it, we have a long non-coding RNA, and its name is Tera, that we have studied this. So in our body, it has designed itself so beautifully that there is everything, and everything is regulated. Even telomerase, which is required for the production of telomere, is also regulated. That particular zist RNA, zist non-coding RNA, which is required for a dosage compensation that we are studying since the last two classes, this is also regulated by another non-coding RNA, which is T6. Although this dosage compensation is important, but more than the required amount of doses compensation can also be a problem. What if inactive despite I mean apart from inactivating one of the X chromosome, it inactivates the other X chromosome also? It can happen. So therefore, to make sure that this over regulation does not happen, this zist is also regulated by another long non-coding RNA, which is T6. Okay, so just to remember, take home message from this class is that everything is required in the body, but in modulation. Every enzyme is required, but in modulation. Every protein, every transcription factor, every RNA, mRNA, every enzyme, everything is metaboloid, everything is required in the body, but in its proper amount and in modulation. Okay, so I hope things are clear with you. Could you understand? Were you able to follow what I'm trying to teach? What I taught today? Could you find your pace? I hope you were able to understand. So now we have this is 10th week. Now we have two more weeks classes, week 11th and week 12th. So week 11th class will not be on Tuesday itself. I will update the timing by discussing with the team, because I have a I'm attending a conference, international conference. So it is coming at the same time. Okay, so I have to decide a date. I will decide a date, and I will notify you. But we'll be covering and completing all the course before 16th of April. So 16th of April will be our last class. 16th of April will be our last class, and in between them we will have one more class. So we have two classes now. Last class will be on 16th April, 7 to 9 as usual. And the next subsequent class, I will discuss with the team, the PM, the NPTEL team, and I will let you know. Okay. So that is it for today. I hope you could understand something from the class. So, okay. Bye bye, everybody. Take care. Prepare well for your for your this exam. Final exams are also coming for this course. And if you have any problem, you can always reach me out. Okay. Okay, then you can leave the class. Hmm. So I'll be sharing the slides with you. Hmm, just okay. Okay, we'll let it be like this. Okay.